Carbohydrates are probably the most abundant and widespread organic substances in nature, made from the elements carbon, hydrogen and oxygen, with their general formula written as this. They are essential constituents of living things, such as in plants making between 60 to 90% of their dry mass. For those of us who are thinking about those rippling abs, it is also our biggest enemy. These molecules provide energy for living organisms. The basic units of these molecules are simple sugars that can join together to make complex sugars. Our first group of carbohydrates are monosaccharides, or simple sugars. These are fast-acting sources of energy and include glucose, fructose, and galactose. The general formula for these molecules is written as this. There are three types of monosaccharides that you will come across during A-level biology. These are triose sugars, pentose sugars, and hexose sugars. For triose sugars, their general formula is C3H6O3. These sugars can be seen in the first stage of respiration, where glucose is broken down into a triose sugar called glyceraldehyde 3-phosphate, or GALP, also known as triose phosphate. For pentose sugars, their general formula is C5H10O5. They are found as ribose and deoxyribose. In DNA, the nucleotide structure is made up of a deoxyribose sugar, and in RNA, this is called a ribose sugar. For hexose sugars, which are sweet tasting sugars found in food, their general formula is C6H12O6. These include glucose, which has two isomers, alpha and beta. The difference between these molecules can be seen in the position of the hydroxyl group bonded to the first carbon. If the hydroxyl group is below the ring, then we call it an alpha glucose. If it's above the ring, we call it a beta glucose. Other hexose sugars include galactose and fructose. Our next category of carbohydrates are disaccharides, formed when two monosaccharides are joined together. Examples include sucrose, made from glucose and fructose. This is a sugar found in plants and is translocated in their phloem, where it moves up and down the plant to where it's needed. Lactose, made from glucose and galactose, is found in milk. And maltose, made from glucose and glucose, is formed from the breakdown of starch and can be extracted from germinating barley and used in brewing and in food manufacturing. The joining of these monosaccharides happens in a condensation reaction. A molecule of water is released during this reaction. The covalent bond holding these two molecules together is known as a glycosidic bond. In maltose and lactose, this is known as a 1,4 glycosidic bond. The 1,4 refers to the position of the covalent bond between the first and the fourth carbon on adjacent sugars. In sucrose, it is a 1,2 glycosidic bond. A disaccharide can be broken down in a hydrolysis reaction, where a water molecule is added to break the glycosidic bond down. Our final group of carbohydrates are polysaccharides. These carbohydrates are made from many monosaccharide units joined together by condensation reactions and are held together by glycosidic bonds, forming a long polymer chain. These molecules can share some of these characteristics. They are insoluble, they are large, they are not sweet, they contain many glycosidic bonds, they have no overall osmotic effect, meaning they do not cause water to move into and out of the cell by osmosis. They are made from more than 2 units. They can be hydrolyzed, usually in the presence of an enzyme. They are very compact, taking up very little space, but have a high energy density. They can be physically and chemically inactive, so they don't interfere with other functions of the cell. Examples of polysaccharides include cellulose, which is found in the plant cell wall. They can't be digested by humans, so act as fiber to create bulk to help muscles of the intestinal wall push food through the digestive system. It is an unbranched, straight-chained polymer formed when beta-glucose molecules join together form 1,4 glycosidic bonds. These cellular chains can pack together into fibers. These bundles of about 2,000 chains are held together by hydrogen bonds and are called microfibrils. These bundles are very strong and are able to resist tension stress. While it can be hydrolyzed to glucose, this only happens with great difficulty and you would need to heat it with concentrated acid to break it down. Our next polysaccharide is starch. This is how glucose is stored in plants. It can be broken down to release energy for the plant. This carbohydrate acts as an important store of energy in plants. While starch is insoluble and compact, it can be readily broken down to release glucose in plants. Foods such as potatoes and bread contain it. It is made up from repeating alpha glucose molecules in a mixture of two polysaccharide chains called amylose and amylopectin. Although amounts vary, amylopectin makes up around 70% of starch. Amylose is made from around 200 to 1500 glucose units, releases glucose more slowly because of its unbranched structure. It is made up of 1,4 glycosidic bonds, and as the chain lengthens, the chain actually spirals into a helix, making it more compact for storage. Amylopectin contains 2,000 to 200,000 glucose units per molecule. Like amylose, it forms a helix, but unlike amylose, it is a branched polymer. The branched chain gives many terminal glucose molecules that can be broken off by hydrolysis to release glucose rapidly. Amylopectin therefore has 1,4 glycosidic bonds and 1,6 glycosidic bonds. Both molecules are not very soluble in water. In plants and animals, amylases catalyze the hydrolysis of starch. Our final polysaccharide is glycogen. Excess glucose in animals is stored as this molecule in the liver and muscles, and it can be broken down to release glucose for respiration. It is also how fungi store glucose. Like starch, it is also very compact and is very similar to amylopectin. It has 1,4 glycosidic bonds and 1,6 glycosidic bonds. However, unlike amylopectin, it is much larger and has more 1,6 glycosidic bonds. The terminal glucose molecules mean that glycogen can be rapidly broken down. It is insoluble in water, and so this hydrolysis needs to happen in the presence of enzymes. Hi, my name is Mr. Science, aka Salim. If you're new to the channel, please remember to like and subscribe, and for more teaching resources, you can visit my website at www.mrscience.co.uk.